Yep, that's no problem. Okay, great. So thank you very much for coming. This is a follow-on to the presentation we did at the Oxford Real Farming Conference. We are the Soil Ecology Laboratory. My name is Daniel, I'm the director, and Adam, who's joining me from the other room, is the bioinocular engineer. And what we're going to be talking about today is a little bit about what we do, and then as much as we can about show you how we use the microscope to assess biology. Mind you, this is as long as we do have 2 hours, but it's not really long enough for you to learn it as a subject matter to be able to do it yourself. So this is, don't treat it as a training session. It's more of a demonstration. But the reason for this webinar is that we didn't have enough time for questions at the Oxford Real Farming Conference. So what we're going to try and do is have the introduction and the microscopy so you can see things, but then leave at least that hour for questions so we can have a discussion so you can learn more. Maybe if you've got some questions, you'll be able to have them answered. I say that, and then basically I have to put in the caveat that we are very happy to say we don't know, and we treat everything here in the lab as an experiment, and we're here to find the truth. And so when we say we don't know, we usually mean that we might be able to speculate, but we don't necessarily have the experience and our own data or people that we're in direct contact with have the data. So that's when that's how we basically delineate our expertise. We try not to talk about things that we haven't tried ourselves, and only extend that to the people that we're in direct contact with. And then if we talk about other pieces of information, then we'll tell you, it's either anecdotal or it's from a paper. So that's basically our approach to this whole subject. So we've got now, wow, close to 40 people on the call. That's great. So first things first, I'm going to share my screen very briefly. This is our website, Soil Ecology Laboratory. So soilecologylab.co.uk. And so this is your best point of contact. The whole story started for me several years ago. I was the first online student here in the UK of Dr. Ingham's school. So many of you know that Dr. Elaine Ingham, she's a pioneer of using not only the academical side, because she is a pioneer from the academic side, but she's also has done a lot of work in the outreach to farmers. And in around 2001, she quit academia altogether and has been working independently with farmers ever since. And so she's got her own website. It's soilfoodweb.com. And we're not affiliated from the perspective of the school, but this is basically where we start from. And we actually try to keep a good relationship with them from the perspective that it's important for us to keep some standards. Direct microscopy, which is taught by Dr. Ingham, was developed by her as a technique for farmers to quickly assess their soils or compost that they work with, to get an understanding with some relatively inexpensive tools of what the biology is doing in the soil, because you can actually spend quite a lot of money on testing, and I will touch on that later. So her work basically started off the real addition to the whole soil biology from the perspective of lab analysis came early in the 80s. I think this is part of her PhD work, actually, where she basically put together the ability to look at all organisms in the soil using the microscope on one slide, whilst especially at the time, the only real methods were colony counts, growing things out on plates, separating things from each other. And so the microscopy, direct microscopy, was a way to do a quick assessment. And the premise of it is that what you see is what you get. So the idea behind the use of microscopy is that if you see something, it's there. If you can't see something, then basically you have to assume that it's not really something that you can expect an effect of. And the reason I say in that way, I'm not saying that it's not there, by the way, because microscopy is not an ideal tool for everything. But there are other methods which will detect chemical signals, and will also detect DNA of organisms that you can't see on the microscope. And therefore, it's a tool that that has been used to assess ecosystems in terms of their fungal, bacterial composition, in terms of the mineralization potential or the nutrient cycling potential with protozoa and nematodes. And to this day, I don't believe other methods have actually repeated those experiments. And so that's why we still use the microscope. It's still a good tool for everybody to quickly assess what's in their soils and compost. The method itself can be used in a scientific manner if you adhere to replication practices. And so if you take simply a single sample and a single drop from a sample and just look at that, then obviously you're only looking at a very small fraction of the volume of the soil or compost. And so to be able to make this a valid tool in an experiment, then you need to look at many samples to be able to assess the results with standard deviations. So you can tell whether basically this what you're looking at, that the numbers that you're getting are reliable. So in general, we were set up to help farmers with transitioning away from agrochemicals and transitioning towards regenerative or organic practices with the help of biology. I got inspired by Dr. Ingham's simple message, which basically says, just make good compost with good biology in it and all of your problems go away. It's a very simple way of putting it, but very difficult to actually implement. And it's something that that is full of anecdotal data from around the world, but there's not enough valid, repeated, replicated controlled trials, and something that is still that has eluded a lot of farmers all across the globe. Actually, there are success stories here in the UK. And they come after many years of trial and error by farmers, and they're usually not a pure biological solution, but a marriage of previous chemical systems and a biological solution. And we're here basically to help simply put those dots together. Spend the money and the labor necessary to be able to, first of all, have a good source of biology to play with, and then once you have that good source of biology, then run controlled trials, which will have oversight of known academic institutions, and provide the reliability and also confidence for farmers looking at these methods. For us as the lab, the end goal is to actually have a product that that they can use straight away, and that the product, if used properly, is going to be able to allow them to switch from agrochemicals to biology in year one, and without loss of yield. And so that's a moonshot. It's a big ask of soils which are quite damaged and denuded of biology in many respects. And so it's something that that we're not there yet. And I'm quite happy to talk about the experiences that we've had with this. So that's in a way of introduction. If you go on the website, one thing that you're going to see here is there's a tab for soil biology testing. And so we have been testing soil and compost for farmers for a couple of years now, more than a couple actually. And one of the things that we did with that data is we created a bit of a top gear leaderboard for farmers who've been sending us samples. And it was quite fun actually. We've put it up here, and farmers were able to look at where they're placed. What we measure using the microscope is 4 main groups of microbes: bacteria, fungi, protozoa, and nematodes. Those are the beneficial microbes that we look for. So the first column is bacteria, the third column is fungi, and then you've got the fifth and sixth columns are the flagellates and amoebae. Those are the protozoa. We're also measuring indicators of not so good conditions, and these would be represented by the overwhelming presence of actinobacteria or not so beneficial fungi, which we would call oomycetes, and then ciliates. And then on the nematode side, you'll be also looking at the presence of root-feeding nematodes or potato cyst nematodes, for instance, things that you don't want to see in the soil. So what you want to see in the soil, in a healthy soil, you would want to see bacteria, you would want to see fungi, you'd want to see amoeba and protozoa. And the idea behind that is when you have bacteria and fungi, they have the access to the total pool of minerals, which plants do not. The way we measure nutrients in the soil is by the means of soluble and exchangeable pools. Soluble pool is basically nutrients that are simply water soluble, and they're the easiest for the plant to pick up. And then the exchangeable pool means that the plant can exchange hydrogen ions with mineral ions that are sitting on a surface of something. And it could be the surface of sand, silt, and clay, or it could be a surface of organic matter. That doesn't matter. As long as there are correct conditions, the plant can exchange simply by putting a little bit of hydrogen into the soil and getting those and releasing those nutrients by just swapping them about. And those two pools are limited, and they're in constant flux. And they're a constant flux when plants are growing, and they're in constant flux when microbes are affecting them as well. But what the plants can't get into is a total pool, and the total pool is actually quite vast. There is a book by Sparks, which is basically soil chemistry book, and the reference is that globally there is no lack of nutrient in the soil for any plant in any conditions. What's lacking is only the access to those nutrients. We even had a conversation recently with a researcher at UK University, and he suggested that we should look at a map specifically made for the UK. And we did, and we ran a calculation basically based on those maps, and those maps were looking at total pools, and they were looking at phosphorus, and they were looking at potassium, and they were looking at many other essential minerals. And we found there is no soil in the UK that is lacking anything, basically. And the conversation was that we should be wary that there could be areas. And we ran the numbers, and at the minimum there were 300 years worth of nutrients in those total pools at minimum. And so the only thing that's standing between those nutrients and the plant are lack of microbes that are doing their jobs. The microbes rely on their food, they rely on the plants. So the plants have specialized in photosynthesis, and so they can take carbon and water and make carbohydrates, sugars, protein, and they will literally send that into the soil, waiting for a response. Plants, when they started evolving about 650 million years ago, terrestrial plants, and they found that basically, sure, we can photosynthesize, but we need other things to be able to perform, all the jobs. We need to create enzymes, this, that, and the other. And so they found that there's this microbial soup, and the microbes had 1.5 billion years of evolution before the terrestrial plants appeared. By the time terrestrial plants, started appearing, there were fungi the size of trees on land. And so here you've got a plant that is trying to make a living in this system. And so it had to get into this symbiosis, basically, where they provide the energy whilst the microbes provide everything else. And so it's a very mutualistic exchange. And so here, basically, using this microscope method, what we're measuring is basically we're measuring that potential. It's not a direct calculation. It's not something that you easily can calculate, basically, and measure that, if you have this amount, that amount of nutrients going to be released, but because you're not going to be able to measure with that very specific laboratory equipment, to have that, exact knowledge of every plant doing everything, because they're doing it whenever they want to do it and they need to do it. So what we basically have is a set of numbers, and that set of numbers comes from studies of Dr. Ingham in the greatest majority, and some of the other researchers that came after her. And the idea is that the minimum bacteria and fungi that we look at is 135 micrograms per gram of soil or per gram of growing medium. And so those are the minimums. And then for protozoa, we're looking at a minimum of 10,000 protozoa, so combined amoebae flagellates per gram or per milliliter of soil or growing medium. And then we would like to have 100 nematodes. So with those numbers, what we're basically getting is that there is a minimum amount of access to that total pool of nutrients, and also a minimum amount of nutrient cycling. So nutrient cycling is bacteria and fungi, they can access those nutrients, but they then become these micro-fertilizer bags, and something has to open them up. And so protozoa, flagellates and amoebae, and nematodes, they're basically acting as the knife that opens the fertilizer bag. So they consume them, but because of the discrepancy between carbon to nitrogen ratios between the bacterium and the protozoa or the nematode, they consume them, but they pee out majority of those micronutrients. They don't need as much as they're actually consuming. They need more of that carbon that they're consuming than all of these other nutrients. And studies were done long ago that basically, if you don't have protozoa and nematodes in the system, then your mineralization rate, or the rate of getting those nutrients which weren't available into the plant, is only about 20%. And if you do have these guys to consume the bacteria, then the mineralization rate is 100%. You get everything that the bacteria basically got stored for themselves, everything can get into the plant. And so it's a self-replenishing system that basically that these microbes operate. In general, from what we've seen, read so far, there are 4 methods for plants to get nutrients through microbiology. And this is one of the more efficient ones by having those nutrient cyclers, having those protozoa and nematodes. So you basically have a full soil food web. So you have bacteria and fungi which collect nutrients from the environment, and then you've got protozoa and nematodes to make these available to plants. So that's one method. Then you've got the rhizophagy cycle, and the rhizophagy cycle is when plants ingest bacteria directly into the roots, and then they hit them with oxygen radicals, open up the cell walls, and consume the contents, literally eat bacteria. And then what's interesting actually is that some of the bacteria actually pass through the roots intact, and they get spit out in a different part of the root, in a different root, and that creates the root hair. So that bacterium now is a source of a new colony in a new area of the root, so that the system can continue. The cycle can continue. So that's the second one. And the third one is endophytes. And part of that rhizophagy cycle is some of these bacteria will actually stay in the plant, either temporarily or permanently. And these guys will perform functions for the plant. They might change one nutrient to another. They might, do something. They might signal something. They might act after the plant dies. They basically sit in the plant and wait for the right signal to do their job. That's endophytes. And then you've got mycorrhizal fungi, which a lot of people have heard about. And mycorrhizal fungi, they come in 3 flavors now. One is the most known, which is arbuscular mycorrhizal fungi, and they create little bushes inside the root cells as the exchange site. And then they grow outside and they gather everything that they want. They can shift things back and forth rapidly. I mean, this has been measured recently, actually, and the actual flow is both ways at the same time, which is mind-boggling. And so there's the arbuscular mycorrhizal fungi. You've got the ectomycorrhizal fungi. So you've got like a sheath of fungi that grow on the surface of the root. It's called a Hartig net, and these guys usually work with trees. And then you've got another version of mycorrhizal fungi which also grow inside the root, and it's actually so new that basically the name escapes me right now. But the intersection is like a little fan, and they are in between root cells, and they act the same way as arbuscular mycorrhizal fungi. They grow inside the root, and they provide that exchange. What's interesting in the difference between the two is that the arbuscular mycorrhizal fungi, they're very good at providing phosphorus to the plant, whilst the other guy is actually very good at providing nitrogen to the plant. And so having both would be ideal. So that's in general basically what the system is. And what we measure is mainly the soil food web. So mainly the system which doesn't require colonization of roots. You can use the microscope to assess colonization of roots by mycorrhizal fungi, but the process is much more involved. You actually have to clean the roots and then boil them for 2 hours in vinegar, and then stain it, and then you're ready to look under the microscope. So it's a very, very involved process. So it's not something that we'll be demonstrating. Right, I'm going to stop sharing, and I'm going to allow Adam to demonstrate how to prepare a sample.
Cool. Do you want to talk through? Well, the window's open and they're shooting outside, so you might have to talk them through it, but if there's no loud bangs, then I'll continue.
No, you're fine.
So we're going to get a slide. That's one of these things. We have a sample of compost. This is a nice fungal compost. Hopefully show you some fungal hyphae. And we have a urine cup with 40 ml of water. You can have a test tube with 4 ml. we use a little bit more volume for the sample. So now I'm going to take one part of our compost or soil that we want to look at and add it to the 4 parts of water.
So if you notice, Adam is using cups instead of test tubes. A lot of labs would be using using test tubes. The reason we use cups is just to increase the amount of sample we're actually putting into that cup. Because with a test tube, you can only put 1 milliliter of compost, and you can imagine basically when you look at that, you know, it's hard to even measure out 1 milliliter, you know, of compost. It's hard to measure out the 10 milliliters. So the reason he's putting it into the water is because we know for a fact that then he gets 10 milliliters of the sample, because the level of water goes from 40 mil to 50. If you were to do it the other way around, you have no clue how much water you're putting in because there's so much space between the different particles.
Cool. So now we're going to shake it for 30 seconds. We have a timer. You can see over here, set to 30 seconds. And we're going to try and make a angular jerky movement. It's going to be like, I'll just demonstrate first, but it's going to be like, can't get any closer because the desk's in the way.
You can move the phone about actually on the head.
Yeah, true, but I'm just going to go for it. I think this demonstrates what we need to do.
Okay.
So each repetition is within a second. I try to do this every time.
Build the cups. We use urine cups, and they are graded to 10 mil. You can buy 20 mil grades, but we grade it to 10 mil because it's then more accurate for us to prepare the sample.
So then we have a homogenized sample.
So the idea behind actually doing this like that every time is because everything can introduce variability. And so even the shaking can introduce variability because this is not a mechanized method. It's a method that requires a technician. It's a method that requires skill. And so what we try to do basically, and this is something that that Dr. Ingham actually teaches through her school. This is everybody basically in the lab went through her school. That's important for us that we have this standardization. And so we adhere to everything that basically allows us to have repeatability of results.
Yeah, and I think it's important to say we also try to be consistent with sampling as well before we even get to this stage. So this is a pipette. And we're going to take we're going to draw from our sample in the kind of center of the water column. It's great to kind of standardize the amount of time from the first mix to the to when you take the sample. So I've vertically dropped a single drop from the pipette, and we'll learn to calibrate the pipettes if we have time. And then we're going to take a cover slip, which is an even more fiddly piece of glass than the previous one. Oh, can barely see it. There you go. I'm going to try and evenly drop this over the sample. I'm going to try and keep the edges of the cover slip parallel to the slide. Usually there'll be some piece of grit that tries to ruin your day. We're looking for even distribution of the debris from the solution under the cover slip. And I like the cover slip to be, yeah, parallel to the slide. So then you can scan through it in a kind of way that means you can evenly see the whole cover slip. To stick together as well. Share screen. See what I see. Now put the sample on the stage. And you should be able to see roughly what we can see through the oculars of the microscope. So to begin with, we just scan for nematodes. They are generally the biggest thing we count. So they're the first thing we count. We do that on 10 times objective, which is 100 times total magnification. For me, it's a little bit out of focus compared to what you guys see.
So at 100 times magnification, what I was looking for is nematodes. So at 100 times magnification, the first thing that we look for basically is nematodes. And so at 100 times we find nematodes, and then we go down to 400 times, and at 400 times we count fungi, we count amoebae and flagellates. And then we want to count bacteria, then we will actually dilute the sample because there's just too much things to look at this dilution rate at the moment. And then basically we would count bacteria, bearing in mind the dilution factor that we have used. I think Adam basically is just trying hard to find something interesting.
Yeah, there's no nematodes in the sample. But maybe you could quickly share the key, Daniel. You got that open in the spreadsheet.
right? So if you see what I'm seeing right now, this is something that you can actually find on Google. If you Google nematode mouthparts, this is something that we use to recognize what nematodes we're looking at. There are 4 major groups that we're interested in. There's the bacterial feeders, fungal feeders, nematodes that attack roots, and also there are nematodes which will eat pretty much anything that they can, and then also there's predatory nematodes, and predatory nematodes actually hunt down other nematodes. So they usually hunt root-feeding nematodes, but they may also be eating beneficial nematodes if the root feeders are not in the area. The ones that you will see most of the time are bacterial-feeding nematodes, and obviously in degraded soils you will find root-feeding nematodes. And these mouthparts are what distinguishes them between each other. There are exceptions to these rules, which sometimes can be quite surprising, but in general, in a bacterial feeding nematode, there is a cylindrical mouth part that you can actually see as almost a little square on the image. And then there's things that we call bulbs. So there's the median or the middle bulb, and the terminal or the end bulb before the rest of the intestinal tract. And so bacterial feeder will have those 2 bulbs, and they're quite prominent in bacterial feeders. And then we've got the fungal feeders. They actually have a little spear, and sometimes they'll have a second spear next to it as like a shark. Basically, it loses one spear, the other one grows in its place. And they'll have a median bulb. They will not have a terminal bulb. Sometimes they'll have a little pin at the end, and we categorize them as switchers. So if there are no fungi in the area, then they might be actually going for plants. There is also, to add to the confusion, there's also a predatory nematode that feeds on fungal feeding nematode that actually has that kind of spear as well. So, but in general, we look at general morphology. The little spear basically gives you an indication that that it consumes fungi, or it gets up to the size of fungi, almost kisses the hyphae, punctures it, and sucks out the inside fluids. The root-feeding nematode, the plant parasitic nematode, has got a different mode of operation. It's got a different spear. It also has a spear, but it's quite thick, and it's got a bulb at the end, so it looks like a pin. And this bulb is necessary to attach muscles, because it actually acts like a jackhammer, and it will literally rip a cell wall open and then slide its entire body in. Adam, are you ready to show something, or are you just waiting for me to finish?
Yeah, we can go back to the slide.
Cool. Right, all yours.
Make it good on your end. Cool. So now this is in our product. This is the goop. It's pretty easy to find a nematode in here. It's actually 2. So this is still at 100 times total magnification. And to identify them, we drop down to 400 times.
So when Adam speaks about speaks about magnification rates, if you look at the microscope, you've got the ocular here, the things that you look through, and then you've got the objective. And the ocular usually is 10 times magnification. So that's what Adam's showing on the other one. And then the objective basically is what you can change. and the ocular times the objective is your total magnification. So this is 100 times total magnification, 10 times ocular, 10 times is in the ocular, and 10 times is in the objective. 10 times 10, 100. The once he changes to 400 times magnification, that means his ocular stayed the same. He changed to a 40 times objective. So now it's 10 times 40 is 400 times magnification. We also have, on the microscopes, we also have 4 times objectives, which you can use when you're setting up the microscope and you just want to have a general view. We actually quite rarely use them, but we still keep them for that. And then you can get, usually microscopes come with a 4th objective. This style of microscope will come with a 4th objective. Usually it's 100 times magnification alone on the objective. So it's total 1000 time magnification. The challenge there is that the air gap between the glass and the objective is already affecting the image that you're getting. And so you use that with a drop of oil. And so first of all, we don't need that kind of magnification when we're identifying organisms in the soil. And second thing is, it's a hassle. You now have to clean that oil every time of your pieces of glass, and you're not really increasing your effectiveness with these organisms. You can identify them at 400 times. And there is also a compromise objective that you need to buy separately usually, and they are the 65 times magnification objectives. And so they actually still work with that air gap, so you don't need oil, and it's already a higher magnification than 400 times, so that if you really need more detail, you can. But again, we just don't use them. We've got one microscope with that objective. We bought it. Frida, who's sitting next to Adam right now, she's got it, I think, on her microscope, but I don't know if she ever uses it because it's not really something that we need. It's not used in any counts. It will be just used to have some fun with the microscope, I guess. I think what's Adam trying to do is find something that doesn't wiggle too much. There is a method of actually slowing them down. There's a couple of methods. One is preparing a sample with koi calm. So you can buy in aquatic shops, you can buy this thing that basically is supposed to calm koi carps. And so this also calms down the nematodes, so they don't wiggle as much. Because if they're wiggling and you go down to 400 times magnification, now it's difficult because now you have to track the mouth parts with the stage, and that is quite tricky. You have to have a very high dexterity and experience to be able to do that, and that's difficult. The other way to do it is quite barbaric, and I'm terrible at it, and that is to use a lighter, and you have to just basically tell them with the heat that conditions are changing. And I think what this is what Adam is trying to show right now. So he's basically showing that if the conditions are changing, then the nematodes can then basically go like, okay, well, I'm just going to stand still. Now, if you do this wrong, like I do it most of the time, see this time Adam was successful. The nematode just slowed down and it's just sitting there not doing anything. But it doesn't look like it's damaged. I mean, once he changes the magnification, we'll actually be able to see whether he's done anything to the nematode, because it's really easy to cook them, really easy. And I don't know how cooked nematodes taste, but basically this is not what you want, because all of the features inside the nematode disappear. They all become one. So what I can see here, you can see actually both the median bulb and terminal bulb. Is that correct, Adam? Is that what you see?
Yeah, it looks a bit different to the ones we usually look at, but definitely a medium bulb and a terminal bulb.
So basically, you can see that there's like a line going in the middle. So that's his digestive tract. Basically, that's basically the esophagus, and the and you've got these swellings. There's one, Adam is pointing to the one that looks like the medium swelling, and then towards the right, there's another one, and that's what we would call the terminal swelling or the terminal bulb. And so that's basically, as that chart was showing, that's now an indication that it's a bacterial feeder. It's a very long bacterial feeder. It's unusually long, actually.
It looks kind of different.
Oftentimes, bacterial feeders, they are half the length of this one.
Now piles, in the young piles, we try to get the levels of nematodes elevated. At the moment, those levels aren't so elevated, so when we extract them into our product, we get a lot more mature adults, and they kind of seem to display the different morphology. They will kind of look very similar when they're juveniles, but when they grow up, they start showing their differences. Cool, so we would manually go through the sample and count these guys. In this sample, I've seen about 3. I haven't done it systematically because we want to move on to talk about the next parts of the sample.
So just to quickly say, you can actually see the entire drop under the microscope at 100 times magnification just by going in a zigzag pattern, one next to the other. It's basically a scanning pattern. And so you can count all nematodes in that drop. And when I was mentioning that for scientific reasons, if you want to make this reliable, what you find is that in the same cup that you just prepared, different drops will contain different numbers of nematodes. And so if you are tasked with being scientific about your soil or compost, then you have to do more than one drop from one sample and then have an average of these nematodes. Normally, we don't do it. It's not really something that that is required to do, especially if you're just doing quick assessments of your samples. But if you have if you're only seeing 3, like in this, for instance, and then you put another drop on the slide and you see none, and you see none, and then you have another drop and you see 8, that's now a statistically very, very different number. And so then you basically look at the averages, and you look at how far from the average are those maximums and minimums. That gives you the confidence in the actual values. There are obviously other methods to assessing numbers of nematodes very accurately. You can use the Behrman funnel method, which basically extracts 99% of all of the nematodes from the sample, and you take like 50 milliliters of the sample to do that. But then you need a different microscope. You need a microscope which actually looks from the bottom of a petri dish, and you have to fill out a petri dish, and then you basically have to have a grid, and you count as many as you can basically on the largest grid as possible. And they're very concentrated. And then basically, if you can count all of them, then basically you have the best results statistically when it comes to nematodes, and you really have the best data. But if you're looking for a quick assessment, again, just direct microscopy using this method. If you have to be more accurate, you just need to do more drops and pull out an average out of those.
Cool. So now after the nematode count, we'll go on to count, well, the main count, which includes fungi, actinobacteria, flagellates, ciliates, and amoeba.
So if you stop sharing for a second, I'll
more.
show the spreadsheet, and everybody will see exactly what we're actually counting and how we're counting that.
Yeah.
So I'll share my screen again. So this spreadsheet, there was a question actually whether the spreadsheet is available. It's available to our students. I am contemplating actually just selling it on the website so that it's more available to others. We've made it simply to be easy for farmers or anybody interested to use. So it actually contains the key within the spreadsheet. So it actually guides the user. Even if you've got a month break, after the training, you can still come back to it, and the spreadsheet guides you through it. So what Adam was referring to, first we did the nematode count. This is the pattern that we've used to look at the nematodes. We've put the nematode value into the window here, and the main count is where we count actinobacteria, fungi, oomycetes, or the fungi that we believe are not good. And I'm going to mention why in a second. And then flagellates, amoebae, and ciliates. That's the main count. And again, we keep a key of general morphology, both for filamentous organisms and for the protozoa. So I'm going to stop sharing now, and over to you, Adam.
Interesting. If everyone saw that key with the beneficial fungi there, do you want to quickly show everyone that again, Daniel?
Yes, 1 second. Can I increase the size as well?
It's a good example of an organism that's easy to show everyone on Zoom.
So the fungi, basically, the way we assess fungi is this is according to Dr. Ingham, and Dr. Ingham had quite a bit of pushback from people in the fungal community on how she decides which fungi is good and which fungi is bad. And there was a very interesting conversation, I believe you can actually find it, between Dr. Ingham and Dr. Mary Cole. And Dr. Mary Cole is a mycologist. And so the two ladies basically had a head-to-head conversation. And they agreed in the end on this kind of classification. So this is not a typical classification of basidiomycetes versus some other fungi that you might find in the family trees, right? In the family tree. So the way Dr. Ingham basically looks at it is whether the fungi has been around long enough to create structure in its hyphae. Because if it's been there long enough, that means that it's somebody who's not opportunistic. It's a fungi that is actually there for the long term, and it's going to be working with others. It's not going to be there just to simply take resources and run. So if a fungi has got color, if a fungi has got thick hyphae, if it's got well-spaced or equally spaced septae, or the division between cell walls, it breaks cleanly with a 90-degree break. And sometimes it's got some added signals, like these little dots looking like a man's stubble, which is basically oxalate crystals, or oxalic acid crystals. And then, you'll be seeing to high heavens when you see clamp connections between cells. That basically means that fungi is there for the long term, and it's been there for a while, and it's been building structure in the soil or on compost. On the other hand, oomycetes is the definition of an opportunistic organism. And the best thing that's going to tell you it's an opportunistic organism is not just looking at the hyphae, but if you can see the whole organism, you will see spores being produced in large quantities, and sometimes zoospores, so the guys that will actually be floating about. And these hyphae are thin. There will be septae, but oftentimes they'll be irregular. They'll be frothy on the inside. The diameter won't be consistent. But the thing that throws us off, and this is where the pushback comes from as well, is that beneficial fungi can look like this, and also not so beneficial fungi could look like this. So the main pushback is from the fact that all young fungal hyphae will look closer to this. You can take wine cap mushrooms, which we use all the time and they're very beneficial. You can look at oyster mushroom, any other gourmet mushroom under the microscope. Freshly growing hyphae will look like this. It's going to be frothy on the inside, it's going to be clear, and it's going to have thin walls. And so it doesn't tell us that it's not a beneficial fungi. So it's only this period of its development that basically means that it's a freshly growing hyphae. So that's something that we like to keep in mind. So if you have a well-developed compost which has been aerobic, which has had lots of woody elements in it, then you should be able, after several months, to start seeing these guys. And that's what you're looking for. And if you're seeing still in that several-month-old compost, if you're still seeing these guys, that means that there's basically some nutrients floating about that are not being taken into bodies of microbes, and opportunistic organisms still have a chance to develop, and they can also then attack your plants. So that's why we're looking at this to gauge how well the compost has been developing, and we're looking for these guys to be present in our solution. We're going to stop sharing now, Adam.
Yep. Yeah, so like Daniel was saying, the oomycetes more indicative of our strategy organisms or opportunistic organisms, while the well-structured, even diameter, even septated fungi is more indicative of K strategy. So equilibrium forming, more indicative of mature systems. And here's an example of that, just like you saw in the key. So we can see this relatively right angular break where the cross wall would be. You see the various cross walls here, and they're relatively evenly spaced.
And also, obviously, the camera is playing with exposure, but the thing is that this hyphae is darker than the background, so it has color. It's not fully transparent.
The sound is kind of unusual. You will see little indentations in the diameter, which will change the overall diameter. That's just where the hyphae was growing around something in the environment. Sample's now drying out, so I'll switch to a different sample.
In the meantime, Katie asked about replicates for nematode accuracy. In science, most scientists will tell you do 3 replicates for standard deviation for them to be happy. So if you're counting nematodes and you want accuracy replication, that you need 3 replicates to be able to do that, so 3 drops. The wording, and you seem to imply from your wording that for all the organisms, all the other microorganisms, you would be content you only using a single drop. And normally, yes, as well. So the spreadsheet actually that we use, and the software that Dr. Ingham's students use, they all calculate standard deviation. And if the standard deviation basically, and standard deviation for all the other organisms is usually enough within 1 drop. And there's a difference between measuring nematodes and measuring other organisms, in a sense that you count nematodes at 100 times magnification and you see the entire drop. And so if you don't see enough in 1 drop, then you go to other drops. But when you're doing this count at 400 times magnification, your field of view is much, much smaller. And so you cannot see the entire, you will not be able to scan the entire slide. You won't be able to count all of the fields of view because there's over 2,000 of them. So now you have to choose only 20 to 25 of them. And you have to be within that number, basically. You have to fit yourself within that number. And so if you don't see enough fungi, for instance, within that number, then you know that your confidence in the number is low. And usually taking more drops, even from the same sample, will not change that. It's just basically how little they are in, across that entire sample. The ones that gives you confidence every time are bacteria. There's just so many of them that you never have a lack of confidence for the numbers that you're actually counting for bacteria. Sorry, Adam.
So good. So I'm doing the main count right now. I've dropped down to 400 times magnification, total magnification. And I've picked a random spot on the cover slip, and I've landed on this piece of fungi. So we can see these cross walls. There's one, just there. And then relatively evenly spaced is another one. And this one would be another cell. And this is a break. So we're pretty happy that this is a beneficial fungi. So we're then going to plot its length against the diameter of my field of view, which you can't see. You're seeing a square, but my field of view is a circle. So then I'm going to plot that length against that, the diameter of that field of view, and then we're going to get a subdivision of that diameter. So we're going to estimate a length for the fungi based on subdivisions of that diameter of the field of view. And to get you used to doing this, we have this cool calibration slide. Switch over to that for a second.
So this calibration slide, by the way, you either get it with a camera when you purchase a microscope, or you buy it separately if it doesn't come automatically with the camera. And it's used to actually calibrate the grid that you see on the screen. But we actually never bother calibrating the software because we don't have to do that. We do everything through the oculars because it's just more accurate to actually look with your eyes than count on the computer screen. I know of a Dr. Ingham's student who has got bad eyes, and he strictly relies on the screen and on well-calibrated grid on the screen. And so you can basically use this to calibrate the grid on the screen. But what Adam basically is showing you is that you can use it for two things. You can first of all, get an idea how wide is your field of view. Now, when you buy a microscope, the person selling you the microscope or the person training on the microscope will tell you what it is. So with this microscope with a 10 by 18 ocular and 40 times magnification objective, the width of that circle that you can't see because you're just seeing a cutout from that circle is 450 micron, which is just under 0.5 millimeter. And so I believe, Adam, you're at 100 times magnification right now because you can see the entire scale. And that entire scale is 1 millimeter across. The entire thing is 1 millimeter across only. So if you were to drop down now to 400 with this scale, now you can see that the distance between the smallest distance between the individual bars from the left-hand side of one bar to the left-hand side of the other bar is 10 micron. So it's 1/100th of a millimeter. So it's a very useful tool actually for learning to assess the width of fungal hyphae, because the spreadsheet basically, or the software Dr. Ingham is using, basically allows you to put in the width of the hyphae, because the width of the hyphae gives you the diameter of the hyphae as a circle, and then the length basically times that circle. Now that's a cylinder. So we're actually having to measure the volume of the cylinder of that hyphae, and then times that by a known. This is just a calibration slide. This is what you're looking at. So basically we're basically having to measure the volume of the diameter of the volume of the hyphae, and then there's a known density of fungal hyphae. So you times the spreadsheet basically times the volume by the density, that gives you the mass. So we're always working in mass of these microbes that way, right? So the interesting bit about this tool is Dr. Ingham basically teaches that you could use simply just the smallest bacterium, and you can try and fit as many of the smallest bacterium, across the hyphae mentally. But that means that basically this is very much up to interpretation. And so what I suggest to anybody who has gone through our lab is, and Elaine actually agrees with me, this is fine with her, is to use this to almost calibrate your eyes. Because when you look at this through the microscope, you get used to the fact that these lines have a width. And when you look at, and when you know that from one side of the line to the same side of the next line is 10 micron, and then you visually divide that by half, that's 5 micron. And within that 5 micron, you can assess the width of that line. And I can tell you that by looking at this, I think it's about 2.5 to 3 micron. I would say 2.5, basically, even looking at the screen of the computer. And so if you basically get used to making samples using a slide like this, then you train your brain to assess the width of the fungal hyphae based on this image. And that makes you more accurate, again, in your measurements. There have been situations where some people were really happy to see fungi. And fungi can be quite challenging for people to grow out in compost. And so when people are happy to see fungi, they will often overestimate what they're seeing because they want to have more of it. And so we've had situations where somebody thought the fungal hyphae was 13 micron in diameter, but in fact it was closer to 4. So it's important to use something like this to have as much standardization between technicians as possible. So this is what we do in the lab. We cross-reference each other that way to be able to do that. Adam, what do you see?
Here's another example, just going back to the sample of even septations, even diameter, dark color, all the things we look for beneficial fungi. And so this, if we imagine it basically covers the field of view, it makes it very easy to count, right? We just count it as one diameter of our field of view. If we were to subdivide that, we would express it in decimals on the sheet. So 0.1 of a diameter, 0.2, 0.25, something like that. That's how we would derive the length. And then looking at the diameter using the bacteria or using our calibrated eyes, using the calibration slide, we'd arrive at a diameter. And we'd input those numbers into the sheet.
The other thing that you,
sorry, go for it.
The other thing that you can see in this image, Adam, if you change the focus, you can see a couple of spores. Now, we don't count spores unless we're asked to specifically. But we do have spore charts basically for our students as well. This is something that's been promoted by Dr. David Johnson, who says quite a lot that in a 12-month-old compost, you don't see any hyphae, you just see spores. Now, we do see a lot of spores. We also see hyphae. Adam, how old do you think this compost is?
This is, yeah, I mean, some of this is really old. It's a fungal compost. It's over 1.5 years old. It's probably, most of it is over 2 years old. And there is some hyphae on the screen.
So this is basically, just another example of making sure that what we're doing is not just attached to a dogma. So we try to test all claims. We don't blindly simply follow and just keep repeating the same things. We like to present other people's views and show our experiences.
Just scanning around, you get an idea of what the environment of compost looks like on the slide. You have these different pieces of organic matter. Like Daniel said, you have spores. Just trying to get that a little bit more focus. You have the mineral fraction. Here is a mineral. You have lots of tiny colloids and bacterial cells. They're just these tiny dots. Some of them look like filaments. Some of them rods, commas, spirals, wiggly lines. And then this guy is an example of a protist in the amoeboid group. So another thing we're looking to count, another thing that eats bacteria and secretes plant available nutrition. It represents our fertilizer liberation powers in the soil. So our mineralization rate could be linked to the population of the things living within the substrate.
So this is what we would call a testate amoeba. And in compost, you see, in aerobic compost, you mainly see these guys. So there are naked amoebae, and then there's testate amoebae. And if compost basically is aerobic and it's not too wet, you're going to see these guys representing, as Adam called them, amoeboids. And so the more mature compost, some other shapes of these testate amoebae appear as well. The naked amoebae, we rarely see them in regular compost, but you can make an experiment. You can take compost and you can wet it out. And when you keep wet compost for a few days, and the other thing you could add to it is some fresh greens, some milled fresh greens basically into the compost, keep it wet, and after a while you would have a plethora of pond organisms, and that includes very large naked amoebae as well. And they're very easy to see. They're quite mobile. They ooze from side to side. And then you will also see a lot of ciliates of various sizes and potentially other organisms as well. But regular compost, which is kept aerobic, usually you'll see these guys, and you usually also see flagellates. And that's basically what you're after.
So this guy, we just count 1. If there was 5 in field of view, we count 5.
So I think it's already
another example of a testate amoeba that looks a little bit different.
So whilst you do that, if you stop sharing, I'll show show something else.
Yeah.
So the Adam is basically looking to find other organisms that would be filled into this count. I can see that basically we're talking a lot, and I want to leave some time for questions. The next section is that you would be looking for bacteria, and this bacterial morphology diagram you can find on Wikipedia. So bacterial morphology diagram, if you search for that. And that's another stage basically in the assessment. And then once the assessment is complete, then basically you have enough information to create an idea of what your soil is capable of supporting. And so, for instance, here, it's just a random set of numbers that I was putting in at some point. And the report basically tells us that the biology in the sample is not suited to grow the desired plants. I'm not saying the sample that we just looked at, because that we didn't put any numbers in. But the idea is that here we're looking to grow top fruit. The target fungal to bacterial biomass ratio was 5, but the sample fungal to bacterial biomass ratio is only 0.46. And so it's mostly suitable for brassicas. So these tools basically allow you to just have that, and all of the information that you've been putting into the spreadsheet or the software that Dr. Ingham School uses basically spits out all of these details. What was the biomass of all of the organisms? What was the confidence in the numbers? What sort of desired range you want to have for any kind of group of plants that you're trying to grow? And then same thing for protozoa, same thing for nematodes. This particular tool, the tool that we've designed for our students, also basically takes those numbers and puts them into words, into English. And so it's just a formula that takes those numbers and translates them, how to understand it. So it says that the biology in the sample is not suited to grow the desired plants. It tells you whether it's bacterial dominated or not, whether there's sufficient numbers to grow the plants, and whether you do see or you don't see any non-beneficial organisms. And the other thing I wanted to show you is that everybody who learns with us also has access to a vast number of recordings and pictures that we have taken over the last 2 or 3 years of various organisms. So the testate amoebae, for instance, that we looked at on Adam's screen, we have come across ones that were actually moving, and this doesn't happen very often. So basically, we've collated this library of images and videos for you to be able to learn quicker. Now, I'm never terribly keen to sell myself. We do have a course and we do sell it, but if you wanted to start on the cheap, first of all, you can go on YouTube and you can find old videos of Dr. Ingham showing how to prepare a sample. You can also go on Facebook and sign up to groups for soil microscopy, for soil food web microscopy. A very good group is also fungal microscopy. Oftentimes, if I don't know what I'm looking at, I will send them a picture of the mushroom and of the spores, and they will tell me exactly what species that is. So there are many free resources that you can go to online to start your journey without spending too much money. The thing that you want to spend some money on is the microscope. And I can tell you now that if you show me a microscope called Swift for £200, I will cry. And do not buy it. It's just something that I spent a day on, and I do not want to spend another day on that microscope. It's not accurate, and it's very dark. Its light is very dark. So so in general, basically, we've got this tool to to see what's in the soil, to see what's in the compost. Do you have enough microbes in the compost to start experimenting with by adding it to your seeds or adding it to your soil? We have used this tool to develop our compost and the product that we sell, as Adam said, the goop. The website where we sell it is down right now for whatever reason. It's crashed today. Sorry, I can't even sell the only product that we sell. And but in general, it's on Soilsmiths and .co.uk. I've just put it in the chat, and it's basically a refined extract of the compost that we make, and it's very useful if you're just learning microscopy. It's useful basically for you to get a small amount because everything is in it, and you can very quickly basically look at identifying all of these microbes without going around and searching for them.
Yeah, for the longest time, you know, when we were starting, we didn't have a material that we could see everything in. So it's
like a reference.
Really good reference resource for that.
Literally
get it ordered to your house. You don't even have to leave the house.
Exactly. No need for trips to Woodlands. Or those are nice. And then in terms of training, if you do think about learning this hands-on and not online somewhere, we're in Hampshire, and there is a training session coming up in the week commencing 27th of February, so it's coming soon, and you can find it at soilecologylab.co.uk. Under shop. Basically, there's microscopy training there. Probably the next training session is going to be towards the end of the year, because we're really not a training outfit. We're here to do R&D. The training session allows us to raise a little bit of money just to keep going, because we rely mostly on investments. And we're actually currently in another investment round. So if you know any people with deep pockets who are looking to invest into early stage companies, we're here to talk about that as well. So the training, basically, just to touch on that, it's a one-day intensive training. You start about 9:30, you finish about 5 o'clock. Usually about 4:30, everybody's brains are fried, and what it prepares you for, it prepares you how to use the microscope. It gives you the tool to record your observations, and it also, the spreadsheet that I was showing you briefly, also gives you a reference to remind you of what to look for and how the process is to be done, so that you don't have to worry that you've forgotten, you know, what you're looking for nematode parts, and it's all in one place. So you can have that morphology reference all in one place, and I find that this is something that I wish I had when I was first starting out. And it's so again, it's just a one day thing, and then you get access to the library, and you really can call us, and you can send us an email, and you know, you can interact with us. We're here. We're trying our best to be there to help. So I think I'm going to open this up to questions. Let me just quickly run through. Oh, sorry, go ahead.
We were on the main count. We'd finish up by taking the sample. Let's say it looks like this, and we dilute it until it looks like that. And then eventually we get to something that looks like this. And then we do our bacterial count. And that would conclude the assessment. Just wanted to touch upon that before we go into questions.
So let me just quickly go because there's some questions that we haven't answered yet. I was talking about the statement about soil nutrients being available, and there was a question about what reference is that, and I'm just going to share my screen. The book is called Environmental Soil Chemistry by Donald Sparks. And on page 60 of the PDF, there's a table, and if you were to look at this table versus what plants actually require, you will find that there is no nutrient that is deficient in the soil, in the total pool. It's just basically the access to it that is the problem. Now, the one nutrient here that basically is going to be a challenge is nitrogen, because it's not something found in rocks for the most part. It's something that's in the atmosphere in abundance, but it's held together, by very strong force of three, a triple bond, and it requires a lot of energy to be split to be able to create ammonia and then ammonium. So there are two ways to do that. One is through the Haber-Bosch process, which requires about 500 megapascals of pressure and pretty much as much temperature in Celsius as well. And that basically requires a lot of energy, and that is something that that is a problem, that is a carbon footprint for agriculture. And the other one is through an enzyme called nitrogenase, which is present in nitrogen-fixing bacteria. And it's present in free-living nitrogen-fixing bacteria, and it's present in bacteria that live with legumes or rhizobia. And so there's also
it's the only way nature is invented.
And there's also a third one called Frankia, which is an actinobacteria, and it lives with in woodland ecosystems with fungi. And the thing is that the current understanding is that the ability of free living microbes is about to fix nitrogen in the soil is about 20 kilograms of nitrogen per hectare. And majority of farmers will use 10 times that to grow a crop with synthetic nitrogen. So they'll apply 200 kilograms of nitrogen per hectare. And the thing is that the reason, for instance, legumes are used in rotations is that in literature you'll find that they can fix between 40 and 160 kilograms per hectare, which is something that is very valuable, and it's very efficient in that regard. But then you have a problem of how do you transfer that legume nitrogen into your wheat? Because then those are two different plants, it's a different system. So this is where having a healthy soil food web where you've got free living nitrogen fixers and fungi working together is important. There's a couple of interesting papers that nobody really talks about. One is a lab experiment where a primary decomposer fungi or oyster mushroom, something that you could eat, was paired up with a rhizobium bacteria that lives in soybeans, and was given a food resource that only the fungi could access. And the fungi fed the bacteria, and the bacteria fixed nitrogen for both of them. It cost the fungi half of the carbon that it was using, but the whole system produced enough nitrogen for both organisms. And then there's another paper which basically the authors grew wheat, and they used the consortium of mycorrhizal fungi, free-living nitrogen fixers, and also some bacillus species for other micronutrients. And that was enough to increase the protein content against the control 5-fold. So this is something that is largely underutilized, and we are taking steps towards using this as a viable tool. So it's something that's still in development, but it's something that we should be looking closer at. I believe there are some questions here now. Please, can you link both these two papers, the oyster mushroom and the wheat protein increase one? I'll try to find that. Yes. If you email me, Katie, I can just email them to you because I've got them on my drive. Anything else? If you want to unmute, go ahead as well. We can have a chat now. It's so quiet.
Is that an Eddie Bailey?
How are you guys cleaning your slides and stuff?
Right. Let me just get the most important piece of kit. This is an ultrasonic cleaner for glasses that you can buy on Amazon for 20 or 30 quid.
They come in many shapes and sizes.
Exactly. And it in 2 minutes, basically, it cleans your pieces of glass. What you need to do then is use a nice clean cotton cloth to wipe them dry so you don't have any smudges. I have failed to complete my project. I was trying to make a nice system where you can just dip them into deionized water to just rinse them off, and then you can just let them dry. But it's basically the simplest way is to use this and then dry them up with a cotton cloth.
Yeah, hi Daniel. Are you still doing your compost league table?
Well, quite happily waiting for some contenders.
All right, okay. Yeah, I had a chat with you at the Oxford Real Farming Conference. Yeah, okay. Yeah, I'm happy to send some in.
Yes.
Is there what's the cost? Is there a cost per sample?
Yes, it's £55 plus VAT.
All right, okay.
The sampling information is on the website on the solidcollegelab.co.uk.
Yeah. Okay, yeah. I'll adhere to it.
Super.
When we send the samples in, you'll give us a report?
Yes, of course.
And sort of, yeah, how good, bad, or indifferent it is in layman's terms.
Yes, we try to basically make it as straightforward as possible and not complicate it with too much jargon, but also just put some pointers to how to understand it.
Yeah, great, thanks.
It can be important to have a word with the technician that conducts the assessment. That can be, you can often get a little bit more from that than just the data alone. It can be kind of useful for all kinds of things like dialing in moisture level in irrigated soils. If things become too anaerobic and waterlogged, you will see ciliates show up. So you start to see these microbial indicators for the state of the environment. And that could help you dial in irrigation, stuff like that. It's also really good for using to test products that say they stimulate microbiology, like a seaweed extract or something like that, or maybe something you made at home. You can apply the product to the soil that contains all the organisms, or to compost that contains all of the organisms. You can do a baseline reading, and then you can follow how those inputs interact with the different microgroups using the bioassay. So that's really something I've found really useful to use it for.
Just basically as a preparation for anybody who is looking to learn the microscope. If you email me, I can send you some links to products that you can use for your little lab setup. I'll just share my screen very quickly. This is the kind of microscope that that you saw them use. So my screen share is loading right now. So hopefully it's going to load in a second. We're not affiliated with Amscope, but it's just something that we use. We've got 7 of these microscopes in the lab, and this is the bare minimum that I would go for. it's interesting that this is obviously, you know, a Chinese production that's been on the market for quite a while. What's interesting is that it's a direct copy of an old Olympus CH4, which was a microscope sold by Olympus in the 80s. And I really like the how the oculars are actually mounted. They're not on a swivel, but they go side to side when you adjust for the width of your eyes. And I found that the swivel can break in some microscopes and they just droop. And here you really don't have a chance of doing that. But this is basically what we would use. It's a trinocular. So you've got two oculars for your eyes and you've got the third ocular for the camera. It's very useful to have the camera because then you can share it on social media. If you're part of groups, you can send it to other people basically and just ask, what is it I'm looking at, you know? So it's very useful to have a camera. It comes with a stage that basically you can manipulate, and it's nice and sturdy. Underneath there's a collector that has a diaphragm which allows you to shade the image so you can actually see the microbes because they're actually transparent, and if you don't shade them, you can't actually see them. So yeah, this is the bare minimum that we would advertise you to use. So I'm going to put that in the chat. We can have a look, and if you email me, basically, I can send you a whole list of other things that you would use to set up your little lab. Julia is asking whether this is compatible with Apple computers. It's a great question, actually. The camera itself is, that I know for sure. Sometimes people have simply used the built-in software for just using the camera on the Mac. The challenges with the software that you saw Adam use on the screen, and a lot of people have reported that it didn't work for them on the Apples. However, Paula, who's also in the room with Adam, I believe we successfully installed it on her MacBook Pro. And so I would say now that yes, it is compatible, fully compatible with Apple computers. So the only thing that you're basically thinking in terms of compatibility is that camera. Obviously, doesn't matter for the microscope.
Can I ask, Daniel, have you ever looked at the effects of biochar as an additive on the biology of a compost or a soil?
No, we haven't yet, actually. We've been so focused on just developing the compost and the biology in the compost that we've not had a chance to play with it.
What's your feeling about its potential role?
That's a very good question. I would expect, first of all, that it would be a means to increase the bacterial biomass in the soil, because it's supposed to act like a little apartment block for the bacteria where they can develop without being predated on by protozoa or nematodes, because they can't get into those little nooks and crannies created by the charring process. But no, we don't have enough experience to comment. We do have some anecdotal data.
Incredibly variable.
Say again, Adam?
I'd imagine it'd be incredibly variable depending on what it's made from and the process and the heats involved and everything like that.
Because this process of feeding the plants, from what I've gathered, depends on the nematodes being able to access the bacteria and convert them into a form that is actually more useful. Have I got that right?
Yeah, so the thing with biochar is that potentially what it can do is that it creates that base amount of bacteria that can then basically keep on reproducing without getting overeaten, overgrazed. Right. And so you've got this overspill, you know, from these basically little apartments. You've got an overspill of bacteria. That's what we've been told the mechanism is. Right. Again, not our experience. So it's something that, and what Adam says in terms of variability is important as well. I've heard people trying to make biochar out of plastic, and so you have to watch out for some things like that as well.
Right. Well, we have a lot of sort of arisings from coppicing and tree work, wood chip, and you know, we tried converting it into you know, a very powdery charcoal, which we'd like to think of as biochar, and I'd be quite interested to see, you know, what the best way of using it would be, whether you would add it to, so our compost comes from cattle manure. So would it be better applied to that and cultured together, or maybe just to put straight on the soil? I'm not really sure yet.
Well, the bio part basically comes from biology, right? So it's not, so obviously when you char the wood, it's empty. So it's, you can think of it basically as a means, you know, okay, I'm going to try and use it to either basically have the most variety of bacteria I can do, and I just put it in straight compost of any kind, or, you know, oh, there's a bug in a jug and I'm just going to want to mix it with that, or, you know, I'm going to want to make kefir out of it and just use it for all sorts of lactobacillus and other EMs maybe, or something like that. So there's it depends really what you charge it with. And again, we don't have experience, so can't comment. If you want to experiment and you know, talk to us about the results, we're more than happy to chat.
Right. Well, that sounds interesting then.
Good. Carolyn's got her hand up.
Yeah, yeah, we're doing a lot with biochar actually over the last year or so through a wholesale plant nursery, because there's a big problem with rooting or quality of rooting of some plants. It's a wholesale nursery, so working with cuttings and with no peat being used anymore, it seems to be a bit of an issue. So we thought biochar might be a solution, and here on the Black Downs we've got a hell of a lot of farmers' brash and hedge clippings. We've been working with researchers from Plymouth University looking at biochar in small spaces as well, because in the nursery it's in trays and pots, and how you can actually increase the microbial activity in those small spaces as well. Now, I bought some of your goop after the Oxford conference, and we've mixed it into, I think, 6 or 7 different mixes. And we've applied for a research project with researchers from Plymouth. There's a microbiologist and a guy who's been doing the Green Wall kind of project over the last 4 years, because we found that actually biochar is good for keeping the resilience of those plants. They're getting less disease, they need less watering, less nutrients. They're all inoculated before going in. And it also has raised the thermal capacity of the wall, which was the reason why they did the project in the first place, was to look at insulation from the outside of a building rather than the inside of a building. So yeah, if we manage to get that project, it could be quite interesting, because we've put goop into the mix as well to see what happens.
That's very interesting. That is very interesting. We're definitely open to projects like that. We're currently, for instance, working as part of a project that's grant funded in the tree industry, in developing a peat-free substitute for tree seedlings, and that's been quite successful actually. They now have plants that grew in the compost that was made early on in phase 1. They now have got plants that grew from seed with 0 fertilizer additions, and they're more than halfway, more than your waist height, healthy trees in a year since planting. So any kind of, you know, research like that is of interest, because what we're just trying to understand how this works in real world. So that we can manipulate it in the shortest amount of time to get the yield results that the farmers need.
There's a really interesting study that was done in Stockholm and urban trees, where they are using just biochar and gravel and a tiny bit of compost, and those trees have grown, I think, 30% quicker and more healthy, and I think they've adopted to plant in that way. And that's about 7 years old, 8 years old, might be a bit more. So, there's a lot of stuff around. So we were just trying to kind of bring everything together and see what we can do for the nursery, but also locally. So we're talking, we've got a lot of anecdotal kind of higher yield vegetables from people who have been using our little starter packs. So we're kind of just...
Well, you need the anecdotal evidence to start with, and then you also need obviously the control trials to be able to validate that. I do things for fun around here. So basically, Adam basically, he tolerates my experiments because he's the main grower who actually knows how to grow things. Him and Paul are great, so I trust them with experiments. But this is something that I do for fun. I took our goop, squeezed out as much water as I could, and I mixed it in to airsoft BB balls. These are plastic airsoft gun balls, something that people shoot at each other at skirmish sites. Okay, and the idea is basically to grow plants in a medium that nobody would care about even thinking of growing plants in. And this is to show that you don't need peat, that you can create mediums with compost, that you can use biology to grow things. And so, I say that this is for shits and giggles, but technical term, by the way, but it's something that really can show potential. There is nothing in this substrate that should allow these plants to grow beyond the seed stage. And this is a follow-on to what I did with tomatoes in play sand from B&Q. And last year I grew tomato plants. I didn't grow them to tomatoes because, again, I thought this was for shits and giggles. And I ended up basically showing that with enough extract in play sand, you can achieve 350% increase in growth over control, because you can't grow in play sand without using it as a hydroponics medium. So yeah, we need to basically have proper experimentation. We need to have visibility of what's happening. Right now, we're starting literally today, finally, a trial in a tent on 3 square meters. We're putting wheat growing with biology against control, using soil from the fields from the outside, because we're on a farm. And we're going to be using microscopy. We're using nutrition analysis by Logan Labs. We're using SAP analysis by NovaCrop. And we're using DNA analysis by Biomakers. There is a full suite of analysis that will allow us as much visibility as we can with a minimum budget. And it still is, it's still, for us, it's still considerable. It's going to cost us probably close to £10,000 before we end this trial. Well, this is going to be the most expensive organic wheat you're going to find in the country, bar none.
Yeah, we've put seeds on for micro veg just in biochar as well. It's grown exactly the same, and much quicker and bigger than samples, and healthier actually. It's completely different.
Very good. It will be, yeah, it's interesting.
We'll be in touch. Yeah, if we
Absolutely.
get the project, it would be great, because it would be really interesting.
Yes, not a problem. Thank you. Any more questions? We've got 5 more minutes. Go ahead.
Good. Just to ask Carolyn if she's got any references to the study, the work that she was just describing.
We've got a Facebook page called the Blackdown Hills Eco Hub, because we're kind of doing it with community. We're setting up a food growing area, which we're going to put biochar kind of test strips in. We're doing some tests on dairy pasture farmland. So we've mixed biochar with slurry as well.
Is that in conjunction with the AOMB?
Yeah, it's no, not really. It's separate. It's with the transition group. So it's transition group, Trim Plants, the nursery, a local builder, and Plymouth University. So we're kind of working together and have been for the last year and a half, 2 years, to see what we can do.
Oh, sounds really exciting. Have you got some funding for it?
That's always the problem. Yeah.
Because you know
well,
that
absolutely no.
AOMBs,
Yeah.
AOMBs have a pot of money from
Yeah.
VEFRA to support
Yeah, it's not very
things
much.
like
so.
that.
Yeah, it's very small amounts. Plymouth University have actually got a funding pot. So this new project is actually coming from the university funding pot.
Hmm.
But yeah, all my time is totally volunteer.
Yeah.
The nursery time is volunteer. Yeah, we're not getting paid anything. So that is a problem because the projects can't really run without people. So I've
That's
managed
what
to
we
get-
find as well. Without people,
Yeah.
they don't just run themselves.
No. We've got funding for things, so like retorts to make the biochar. Yeah, we had giant radishes as well, actually, last
So
year. Huge.
this is actually from a friend of ours, also a student of Elaine's. Her website is livingsoilmatters.com. She bought some goop from us, and she put radishes into a Saturn compost mix with a little bit of goop. Basically, there was can't remember how many milliliters she put into her pots, and she got a 55% increase against control in weight. And so I decided to replicate it. And last year, just before Christmas, I did the harvest, and I got a 22% increase. And that's after putting 10 milliliters of goop into 75-liter pots. So tiny amounts in comparison with any
application.
single application. Yeah, exactly. And then the Soulsmiths website is still down, but you can find Soulsmiths UK on Facebook. And here we put things on it as well. This is the replication of that radish trial. But this is also interesting. Adam finished his chilies in January, and that took him a year, right? So
Yeah,
this
year
is
long.
this is the result from Adam. Adam, you were wanted to prove that seed treatment with biology makes no sense, right?
Yeah, I always thought that, people say with the compost extract that they see a benefit in treatment to seed. And I thought, well, if you for the rest of the life of the plants, you treated them all the same, so they all got exposure to biocomplete compost extract for their whole life, would the single treatment to the seed or into the module make much of a difference? I thought it would make a negligible difference. But in the dry weight of the chilies in my experiment, it made a big difference. So we saw a 47 point something uplift against the control.
So that was the result, basically 137 grams of dried chilies versus 93. So it was about 47% difference. And the thing is, as Adam says, everything else was the same, and both had biology, both had compost, both had extracts. The difference only came from coating seeds. And that is something that we still don't understand. When I was chatting with Professor Duncan Cameron from Manchester University, he suggested that there are substances that if you provide these substances to the seed, the legacy effects resides, show throughout the entire lifespan of the plant. And so he says that we did well. That with the compost, basically we created the situation where we've got these substances. We still don't know what these substances are, and how how they're being applied to on the seeds, whether it's through the action of microbes or already present in the extract through the metabolism that has occurred during the composting process. But basically, these are substances which are not harmful to anybody, whilst industrial alternatives could be actually harmful to human health when they get back into the seed once you harvest the crop. So we got a pat on the back for that recently, which is nice.
That reminds me of a talk that I heard from Walter Jenner about growing crops in dryland, arid situations, where he developed a system for just coating the seeds with some kind of organic preparation. I can't remember what it was, but you know, there may be something from him that would corroborate that concept, I think, Tony.
By the way, yesterday was Valentine's, so happy late Valentine's. Just a couple of nematodes under scanning electron microscope for you.
Thank you.
Our friends at EcoThrive have created a compost blend from their own worm casting and our compost, and this is something that they sell to small growers as well. They're a great ethical company as well. Can't sell them enough. They're lovely friends. Gareth has been in the game for over a decade, and he's very knowledgeable. So we love them lots. So that's why we've put them on our social media as well. There's quite a few really nice people, obviously, who are trying to. Oh, this finally found it. Yes, these are my. If it opens. Hello. On this. There we go. These were my Plaisance tomatoes. Obviously, you can see deficiencies in them, but you can also see the difference in the height. So B&Q Plaisance on the right-hand side with nothing, on the left-hand side with compost extracts of good quality. So that's what. And again, this is not huge amounts. So this is why basically we're doing these things. We're trying to create this alternative so that it's cost effective, it's efficient, and it's available. Because at the moment it's just people can do this on their own, and again we're quite happy to support that. It's just that the majority of the farming industry will simply not touch it with a barge pole because it's complex and it's unproven. And if we don't change that, nothing will change. We'll just continue having the same issues with nutrient runoffs. We're going to have the same issues with carbon footprint of the fertilizer industry. So there's there's definitely a great opportunity. Any more questions before we finish? I'd be really interested. Oh, there we go. Holly says, I'd be really interested to know more about Adam's seed coat trials. There's research from Dr. James White and others about the plant microbiome being vectored on the seed coat. I wonder if the inoculant interacted with existing biology in the seed and enabled healthier growth. Just speculating. Yeah, this is the thing. That kind of speculation would require a bit of money to set up proper experimentation. Adam?
Yeah, all I did was put the seed in the module tray. So I put the seed in the substrate, like a horticultural substrate. And then the first treatment, or the first liquid that hit that module tray was an algal goop diluted in water. I used 40 ml per liter of water. So yeah, replicate it, get in touch with us.
I've heard that, Josie says, I've heard that vermicompost in the seed mix affects the life of the plant for health. Affects the health of the plant for life? Is vermicompost, if you think about vermicompost, yes. Vermicompost basically is called composting methods with composting worms, right? And their gut microbiome is basically serves as an inoculation zone for their castings. So what you're getting basically is you're getting decomposition and you're getting re-inoculation with a healthy microbiome. You might want to start with a good microbiome for the beds that you're building. You might also want to create a system where you have, say, a hot compost, and then you put it into, and you actually feed that to the worms. Castings are also very well processed, and so they have, our goop pretty much have got the structure of worm castings. So they are, it's almost like this perfect soil kind of mixture, very small, fine particle size of organic matter, and then microbes on top of that. So you've got a lot of exchange sites, you've got some substances, you've already got some minerals on it. And depending on what you feed the worms, you might actually have a nitrogen content in it as well. So it really depends a lot on what these worms are being fed on. Genevieve asks, what would be the difference between your group and, say, tea made with alpaca poo and nettles, docans, etc.? These sound to me like anaerobic teas. So compost that we make is aerobic. And so anything that would pop out of the gut of a grazier, that would come from an anaerobic environment. So it's a completely different ecosystem. And so a tea made of that, I would say, first of all, something made out of poo, that would be a good extract of nitrogen. You would have a nitrogen source in the form of urea, maybe a bit of ammonia in it, ammonium. So the thing is that it's going to be much, much different. What we actually, what the goop is an aged compost, and we extract basically the smallest fraction in terms of the size. And we find that it's actually quite balanced minerally. We have done some testing, and we're trying to do more testing on that side of things. And also, say again, please.
Not just balance, but the availability of the trace minerals is also really elevated compared to, you know, any native soil.
And then obviously it also contains the microbes that can provide nutrient cycling to plants. Emma has got her hand up. Sorry, I don't want to keep you too long.
Hi, I was just wondering how far along you have maybe got with pairing your microscope assays with genetic testing.
Yeah, this is a great question. It's hard to find much correlation there, because you're going to have everything on the DNA list, and the DNA list is like ketchup. The DNA list basically will give you everything that the sequencer will find. But the microscope will only show you morphology of what's there and then. And you will not be able to speciate pretty much 99% of anything under the microscope at all. Because for every group of microbes that you are actually looking at in the microscope, you would have to have a specialist to have even a chance to speciate based on just morphology.
Yeah, I was just wondering if I got a bit of funding. I'm currently trying to set up an Oxford nanopore experiment to do soil metagenomics. So I'm currently going through teaching myself how to try and set that up. I was wondering, for yourselves, are you looking into that sort of going through for the Oxford nanopore? I haven't. I have been pushing back the delivery of it for about 6 or 7 months because I've been running.
If you talk to me, we actually run some calculations on the costings of all of the equipment and all of the reagents. So I've got a spreadsheet with that.
Oh, that would be amazing. Maybe if we could set up a call, because that's what's been delaying me. Just
email me, Daniel at soilecology.lab.co.uk. I mean, we spent with Frida, we spent way too many days actually trying to work it out, because we tried to make it. We thought about making this as a service because we wanted to look at DNA ourselves. We had issues with reliability with university work in terms of the delays that we were experiencing. So we've looked at basically getting that Oxford nanopore. We finally settled on working with biome makers. So these are the people that we're working with right now. We're just starting, by the way. First sample to them literally went out today.
Whereabouts is the biomakers? Are they based?
They've got 2 labs. No, they've got two labs. One is in the States and one is in Spain. So we're sending samples to
Okay.
the one in Spain. And most probably, I'm saying that, but I'm waiting for results. Once we get some experience with them, I will be able to maybe offer to the rest of the public to be a reseller for them, because what we think, looking at the report that they provide for farmers, might be actually a useful tool for farmers, because it's not just a spit out of a long list, it's actually a list of functions.
Yeah,
for
formatting
Yeah,
is super
that's
important.
perfect.
The formatting of the data you get is probably the most important thing, or it kind of, you know, it makes it more useful. If a layman can look at the data that you receive and extract some kind of, you know, some kind of direction from it, like, is it making soil health parameters? Is it, you know, is management increasing soil health parameters, or are those parameters declining? I think that's the kind of tool that's useful to horticulture and agriculture.
So
Oh,
in terms
yeah,
of you know,
accessibility,
what you get, for instance, from Oxford Nanopore, right? For everybody here, basically, what we're talking about is we're talking about a cheap DNA analyzer that you can actually connect to your laptop, and a system, an ecosystem, basically, that allows you to start with DNA analysis for the lowest possible price that is available on the market. The analyzer itself costs £1,000, and then basically, you know, that's cheap in comparison to Illumina, which costs £60,000. So the barrier of entry is lower. So that's why, you know, there's now these analyzers being used in places in Africa and India, where basically, you know, the cost of analysis can be quite prohibitive. And the challenge with that is that you have to set up a whole lab. Basically, it's very difficult to do this in the field. There are very many steps involved in preparation of samples for DNA analysis, then once you even run the whole process of reading the genes, then you have to be an IT specialist with some knowledge of Python, so that you can go through the bioinformatics procedure just to get the list of microbes. And then you've got this massive list of microbes from the ones that you actually identified, because oftentimes 2/3 are unidentified. Nobody has ever given the names. So now you've got a massive list of microbes, and then you need a person who doesn't have a life. Here in this lab, it's called Adam Swan, to read this over the weekend, basically, instead of spending time with your loved ones. You're welcome. And only then you can glean some information. And so we've basically looked at different options. We have worked with Sheffield Uni, we have worked with FERA, we have spoke with Nature Metrics, and we've spoken with biomakers. And so far, the best option for us that we can see is biomakers, because basically it actually shows some more information than just the raw data.
Another thing we've seen is that the active biomass, as identified on the microscope, does not give you any kind of indication of the diversity of stuff, you know, as identified using metagenomics.
Yes, this is another thing, actually, in terms of the differences. The typical report from bioinformatics, you know, after the run on DNA scanners, is that they give you families, they give you groups, they give you diversity score, and then they frown upon you when you've got a low diversity score. But then, you know, you compare that to, for instance, to a very healthy compost pile, which had so much fungi that nobody has, you know, seen this in this country, and you could grow the biggest strawberries on it. And how do you compare the two? It's two different viewpoints. We're using different tools to get different viewpoints on the same on the same sample. We don't want to be using the same viewpoint all the time. We want to have different perspectives. So we're not necessarily worried about the fact that they're not correlated, because they're giving us two different kinds of information. In terms of correlation, the things that people compare microscopy to is phospholipid fatty acid analysis, for instance, and that is the analysis of the cell walls of bacteria and the cell walls of fungi. That's basically the structure of the chemistry of the cell walls. And based on that chemistry, you can define whether that's bacterium or if it's fungi. And you can also assess biomass. So you're assessing the same thing. However, you can have a completely different result. And so I've had samples that were analyzed with PLFA, with the microbiometer, and I analyzed the same samples, and I've got two completely different results. And because at the time, microbiometer was using the same tables of interpretation as were created with a microscope, I said, don't use it unless new tables of interpretation are created. So they made their own, which are relative to the data set they created. But we're looking at the same thing. We're just looking at biomass of fungi and bacteria, basically, and its and how it relates to each other, which is important for the ecosystem progression. But we're not seeing any other sides of the view. So that's why we look at the chemistry, we look at the microscopy, we look at plant sap analysis, and we look at DNA, because they're completely different viewpoints at the same sample. And that's how we're trying to put this composite view of something that's completely non-correlated.
You start getting into the DNA as well, you start to see things that you can't see with microscopy, right? So you see fungal to fungal ratios, you see bacteria to bacteria ratios that change as ecology develops. So it becomes a little bit different to the ratios of groups to groups. Now you're like zooming into groups, into the fungi, you're looking at differences in the class level, in the phylum level, and eventually the genus levels, and sometimes you get species, but not so much.
Yeah, that's really interesting. Thanks so much. What was your email again, Daniel? Just Daniel@soil.
I just put it into the chat because I think we've run over quite a lot. People obviously are looking to go about their evenings, and we've got another call at 4:30. So is there any more questions before we finish?
It's definitely worth us having a long chat about the DNA stuff to inform your investigation.
Yeah, definitely.
Yeah.
Get in touch.
That would be amazing. Thank you so much for your time.
Yeah, no worries.
No worries.
Anything else?
Thank you very much, guys.
Super. Thank you very much for having us. And I'll make this recording available on my YouTube channel. Just look for my full name, Daniel Tirkiel, and you'll find it. And I'll try to, if there's a list, then basically I'll forward this to everyone if you need it for reference. So thank you very much, and see you some other time.
Okay, thank you. Thank you. I've got you. I have to leave now.